Hello students, this is Dr. Abrar and this is the very first lecture on hemoglobin. So let's start off with a video.
You breathe in about 17,000 times per day. It's a process you rarely think about, but behind the scenes, a huge coordinated effort is playing out. Your vital organs, the gut, brain, bones, lungs, blood, and heart, work together to sustain your life by delivering oxygen to tissues throughout your body. Most of our cells need oxygen because it's one of the key ingredients to aerobic respiration. That's the process that produces a molecule called ATP, which our cells use to power their many incredible functions. But getting oxygen throughout our bodies is a surprisingly difficult task. Gas enters cells by diffusing in from their surroundings, and that only happens efficiently over tiny distances. So for oxygen to reach the cells within our bodies, it needs a transportation network. This is where our 20 trillion red blood cells come in. Each one contains about 270 million oxygen-binding molecules of hemoglobin, which is what gives the blood its scarlet hue.
So what is hemoglobin? It is a hemoprotein whose primary function is to transport oxygen from the lungs to the body tissue. The structure of hemoglobin, it consists of 4 heme molecules covalently linked to two pairs of polypeptide chains. So from its name, it consists from heme and globin and may carry up to 4 molecules of oxygen. So let's take a closer look on the structure of hemoglobin. This is the 2 pairs of the globin chain, designed as an alpha 1, alpha 2, and non-alpha chains. So this is the alpha chains and non-alpha chains. And in each of these chains, we have a heme group. This is the heme group. This is 1, 2, 3, 4. And in the center of this heme, we have an iron ion, which is in the ferrous oxidation state. And here the oxygen will bind with this iron. Okay, so each molecule of hemoglobin binds to 4 molecules of oxygen. Normal hemoglobin types in adults. The main type of hemoglobin is HbA1. It consists from 2 pairs of globin chains, alpha and beta. So 2 alpha and 2 beta. And this type is constitutes about 97% of total hemoglobin. The other type of hemoglobin, which is also found in adults, but in smaller amounts, is called HbA2. It consists from 2 alpha and 2 delta, and constitutes about 2%. Other normal types of hemoglobins called HbF. F refers to fetal hemoglobin, consists from 2 alpha and 2 gamma chains. This type of hemoglobin is present primarily in embryonic life, and it usually disappears from the circulation by the age of 6 months. This is an important information. I want you to pay attention to this. So by the age of 6 months, the HbF will be replaced by HbA, and HbF remain only less than 1% of hemoglobin in adults. So we have 3 types in adults. We have HbA, which is 97%, HbA2, it's only 2%, and HbF, which is less than 1%. The physiological role of hemoglobin, like I said, the iron in heme is normally in the ferrous oxidation state, Fe2+, and it's able to act as the oxygen-carrying entity by combining reversibly with oxygen. There is a term called cooperativity, and this used to describe the interaction of the globin chains in such a way that oxygenation of only one heme group enhances the probability of oxygenation of another or all heme groups. So, as heme takes up O2, there is a rearrangement at the other subunits that allows and facilitates the uptake of more O2. And this is called cooperativity. This rearrangement called cooperativity. The cooperative binding process of hemoglobin is highlighted in the oxygen dissociation curve. This is the oxygen dissociation curve. This represents the joining of the first oxygen molecule will be slow, and the joining of the second and third oxygen molecules is faster, and joining of the fourth molecule is more difficult. Now, the biosynthesis of hemoglobin. It requires the biosynthesis of both heme and the globin polypeptide chains. The globin synthesis, the genes coding for the alpha globin are found as two genes on chromosome 16. Two genes on chromosome 16, while the genes coding for the non-alpha globin are found on chromosome 11. One gene for the beta globin, one gene for the delta globin, and two genes encode for the gamma globin chain. The biosynthesis of heme occurs mainly in the bone marrow and the liver. In the bone marrow, about 70 to 80% of heme will be incorporated into the synthesis of hemoglobin, while in the liver, about 15% of heme will be incorporated into the synthesis of cytochrome. Now, the process of heme synthesis happens both within the mitochondria and the cytosol, and it requires multiple enzymes to catalyze the numerous steps. I'm going to go through each step of the pathway alone, and then we're going to look at the pathway as a whole. The first step of the pathway occurs in the mitochondria, and it starts by simple compound from succinyl-CoA. Succinyl-CoA will combine with glycine, and the combining of these two molecules will produce aminolevulinic acid. This reaction is catalyzed by the enzyme called aminolevulinic acid synthase, or ALA synthase. And like I said, this occurs in the mitochondria. This is a rate-limiting step in the synthetic pathway, and it's regulated by a feedback inhibition by heme. So heme will inhibit its own synthesis, which makes sense because if heme is already available, why make more heme? I'm already self-sufficient. This enzyme, ALA synthase, requires pyridoxal phosphate as a cofactor. And pyridoxal phosphate is vitamin B6. So in case of vitamin B6 deficiency, this will produce problems in the heme synthesis pathway. Now, the condensation of two aminolevulinic acids yields porphobilinogen. So 2-aminolevulinic acid will be producing porphobilinogen by the enzyme porphobilinogen synthase. This reaction occurs in the cytosol. This enzyme, porphobilinogen synthase, will be inhibited by lead. And that's why lead poisoning will lead to anemia, because lead will inhibit porphobilinogen synthase. This will lead to inhibition of the heme synthesis pathway and will produce anemia. From there, 4 porphobilinogens combine to form hydroxymethylbiolane. 4 porphobilinogens will produce hydroxymethylbiolane by the help of the enzyme called porphobilinogen deaminase. And this reaction also occurs in the cytosol. Hydroxymethylbiolane will be converted to uroporphyrogen 3. Uroporphyrogen 3. This is catalyzed by the enzyme uroporphyrogen 3 synthase. This also occurs in the cytosol. Uroporphyrogen 3 will be converted into cuproporphyrogen 3 by the enzyme uroporphyrogen 3 decarboxylase. This also occurs in the cytosol. From there, cuproporphyrogen 3 will be brought back to the mitochondria and be converted into protoporphyrogen 9 by the enzyme cuproporphyrogen oxidase. And it's in the mitochondria. Then, protoporphyrogen 9 will be converted to protoporphyrin 9 by the enzyme protoporphyrogen oxidase. And this reaction occurs in the mitochondria. Lastly, protoporphyrin 9 will be converted to heme, which is the end product, by adding iron by the enzyme ferrochelatase. And this is happening in the mitochondria. Lead also inhibits ferrochelatase enzyme and will lead to anemia. So lead will inhibit two enzymes in the heme synthesis pathway. It will inhibit the ferrochelatase and porphobilinogen synthase. Now this is the pathway as a whole. We have steps occurs in the mitochondria, and we have steps occurs in the cytosol. Thank you.